update. Basically, we are going to cover renal today, and then tomorrow we will start off with the endocrine system. I haven't posted those lectures yet, but I will be probably later today. All right. Oops. No, I didn't want to do that. Let's see. All right. So last time, just to briefly go over, we spoke about the anatomy of the kidney, spoke about the basic functions in general terms, filtration, reabsorption, and secretion, which are three steps that involve processing of the filtrate before the urine is excreted. We did speak about the different regions. We spoke about the renal corpuscle, the glomerular capsule, which is where filtration occurs. We spoke about kind of what's involved with that, the pressures, back and forth pressures, the opposing pressures that determine what the filtration rate is going to be. We spoke about GFR as a very effective way of measuring how much filtration is occurring. So we spoke a lot about filtration. Today we're going to start off talking about reabsorption. And then we'll talk a little bit, not as much, about secretion, pH balance, and then we'll end up talking about the process of micturition, which is really the process of urination. All right. So what is reabsorption? Well, reabsorption is essentially the movement of the filtrate, or like the filtered plasma, from the tubules. You learned about the tubules, the proximal tubules, distal tubules, things like that. Movement of that filtrate from the tubules back into the capillaries. And as I mentioned before, and I'll mention again as we go along, roughly 99% of what's filtered into the kidneys is reabsorbed. Most of the reabsorption, even though we've got several regions of the kidney, right, with the renal corpuscle being where filtration is occurring, and then we've got the tubules, even though reabsorption can occur in any of those tubules, the majority of it occurs in the proximal region, proximal tubules. And it's not regulated, it just happens, just naturally. We will talk about different factors that can influence it though. All right. So what is reabsorbed and what is not reabsorbed? This is a small table, but hopefully you can, excuse me for scratching my nose here, allergies. If we take a look at many of these substances, starting off with water, we take a look at what the filtration rate of water is, what the reabsorption rate is, and what the percent that's reabsorbed. I don't want you to pay attention too much about the specifics of these numbers per se, other than the fact we do see this 180 liters per day for water, but I want you to take a look at the end game. So we've got all these different substances, glucose, urea, sodium, potassium, right? And they're all filtered. At the very end, this last column, note how many of them and the amount of reabsorption that takes place. Except for urea, everything is either 99% reabsorbed or 100% reabsorbed. That would be glucose, right? Urea is the only one that's about 50%. So again, the whole idea is to conserve those minerals, conserve those nutrients, and not allow them to be excreted. Remember, we are land animals, right? So we don't want to lose any more water than we absolutely need to. All right. So here's a drawing of the nephron. If you remember the anatomy of the nephron, right here is the capsule. This is the proximal tubule. This is the loop of Henle where most of the concentration occurs. Here's the distal tubule, and here's the collecting duct, which eventually will lead into the ureter and the bladder. Where these arrows are, we're indicating different processes. This red arrow with the F indicating, of course, that's where filtration is occurring. And as you know, solutes, glucose, amino acids, water all get filtered, but cells and proteins do not, right? And so they stay in the blood. Of all those substances that are filtered, 99% get reabsorbed, and you can see the reabsorptions occurring quite extensively in the proximal tubule, but then you can also see in the loop of Henle, in distal tubule, and collecting duct. But again, mostly in the proximal tubule. Same thing with secretion. Secretion occurs basically throughout as well. And of course, finally, excretion, which is really the end game after we subtract out reabsorption from filtration and secretion, we end up with the volume that's going to be excreted. All right. So I'm going to introduce to you some terms which really can also assess different aspects of renal function. I spoke to you about GFR. Remember, glomerular filtration rate? That's the velocity, or that's the rate that substances are filtered from the blood into the tubules across that filtration membrane. Another term I want to introduce to you is renal threshold. And the renal threshold, kind of ignore this graph for now. The renal threshold is the plasma concentration level above which all of a filtered substance is not reabsorbed in the blood. And I put, for an example, glucose. So what does this mean? So every substance, right, has a certain threshold, certain renal threshold, that the kidney can only reabsorb so much. And if the concentration of that solute is above that threshold, then instead of being reabsorbed, it's going to spill over and stay in the urine. Okay, this is kind of comparable to a dam. If you think about a dam which holds back water, when the water level gets too high, of course, the water spills over the dam, right? If glucose levels get too high in the blood, instead of it all being reabsorbed back, some of it's going to spill out into the urine. So if you get above the renal threshold, some's going to spill out in the urine. And just from a clinical example, someone who has diabetes, say it's not being treated, glucose, as you know, one of the characteristics of diabetes is hyperglycemia, high blood sugar levels. Blood sugar levels get high. The levels in the blood of a typical diabetic are so high that once the glucose goes into the tubules, some of that glucose gets reabsorbed, but not all of it can. And so it spills out in the urine. That's why one of the markers for diabetes is sugar in the urine. This graph is a glucose tolerance test. And if any of you guys have ever had one of these, it's kind of a way of assessing someone if it's suspected they might be borderline diabetic or pre-diabetic. We're looking all these different parts of the graph, these lines indicate blood sugar levels after somebody ingested a known quantity of sugar. It's usually like a sugary solution, all right? This right here is a healthy individual that time zero, of course, is when they ingested the glucose. And as you might expect on the y-axis, the blood glucose levels go up, right? Passes through the gut, goes into the blood. But then at a certain level, typically within an hour, blood sugar levels end up going down and essentially kind of bottom out at about 2 hours. And this is due, of course, to a hormone you've heard of called insulin. We'll talk more about that tomorrow or Thursday. This is someone who's diabetic. And first of all, their resting, or their blood sugar levels prior to ingesting glucose are already high, 150. Normally your blood sugar levels should be, when you're fasting, less than 100, ideally around 70, but this is a diabetic, and you can see what happens. They ingested glucose, and it goes up, 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 peaks at 1 hour, and it goes down, but look how long it takes for the blood sugar to reach the baseline. It takes about 5 hours. Okay, so again, this is just kind of an interesting way of looking at blood sugar, how we handle it. Now, how is blood sugar handled from a molecular aspect or physiological aspect? How does sugar, which is filtered into the kidney tubules, how does it get back into the blood? Well, this happens to be a kidney cell. This right here would be the lumen. This is where urine is going, right? This is the kidney cell. This is the kind of the interstitial fluid, and this is the capillary, right? So we're looking at glucose reabsorption. So we're going from the tubules into the blood. So the arrow is going this way. And you can see right here, glucose is reabsorbed from the kidney tubule into a kidney cell by way of a protein called SGLT, which stands for sodium glucose linked transporter. Raji. Okay. Now it's called sodium glucose linked transporter because both sodium and glucose are brought in simultaneously. Sodium ends up going down its gradient, glucose up its gradient. But nonetheless, this is how it enters into the cell by SGLT. Once it gets into the cell, we still have to get into the blood. And glucose enters into the blood through what's called facilitated diffusion, whereas SGLT is called a secondary active transport. Facilitated diffusion is where glucose goes from this area of high concentration to low, passes through these fenestrations in the capillary cells, between the capillary cells, and goes into the blood. All right, so there's 2 steps for glucose to get back into the blood. Clinically, it's been interesting. One interesting treatment for diabetes has been these drugs that are called SGLT inhibitors, which effectively block this inhibitor so it can't bring glucose back into the blood, so the glucose is excreted, and this helps to overcome hyperglycemia. There's a lot of different treatments, different forms of diabetes, which we'll get to later. All right, so SGLT, first step from the lumen into the cell, and then GLUT. This is called GLUT. It's another transport protein that moves glucose from the cell into the blood. We'll talk a bit about sodium later on. I just want to kind of focus on glucose for now. All right. So I've mentioned renal threshold. I want to mention another term called transport maximum. And this refers to the rate of transport of a solute when the carriers are saturated. What I mean by saturated, kind of like, if hemoglobin is saturated, it's binding to 4 oxygen molecules. That would be 100% saturated. That's 100% saturated. If all the SGLT proteins that are lining the kidney on that membrane all have glucose on them, that's saturated, and it can't handle anymore. So the renal threshold refers to, of course, the level above a certain concentration level in which too much of a solute spills over in the urine, and that's because the carrier proteins are saturated. So it all works together. Why does glucose end up in the urine in diabetics? Is because there's more glucose than there are carriers to handle it. All right. So if a solute, and I'll just say glucose, saturates the carriers, then there's going to be some solute that's excreted. The solute in the plasma that causes the solute in the filtrate to saturate carriers and spill over in the urine, that's the renal threshold. Okay, so, and there is a definite defined concentration, what we call a renal threshold for glucose, in which if the levels get above this specific number, which we'll talk about, it's about like 180. If it gets higher than that, then it'll spill over the urine because that's too much for the carriers to handle. Okay. So just to kind of go over this again, since it is some new information, glucose reabsorption involves a couple things. First of all, glucose has to be filtered, right? It's filtered into the tubules across the glomerulus. and it's 100% reabsorbed into the proximal tubules. Under normal circumstances, no glucose appears in the urine. Like I say, normal circumstances, right? Unless someone's diabetic. The carrier proteins for glucose reabsorption are SGLT, which is on what we call the apical membrane. That's the membrane that's closest to the kidney lumen, and that's secondary active transport. And then we have the basolateral membrane of the cell, which is adjacent to where the capillaries are, and that's where facilitated diffusion exists due to the presence of a transport protein called GLUT. All right. So let's put some of this together and kind of apply it to a graph, and I think this visual aspect is very effective, hopefully, for you to see what's going on here. First of all, on the x-axis, this is showing the plasma levels of glucose. So how much glucose is in the blood? On the y-axis, this is the rate of glucose movement in through the kidney tubules. We've got three distinct lines. We've got this blue line, which is indicating filtration. And as you can see, as the glucose levels go up, there's a linear increase in the amount of it that moves into the kidney tubules, right? Very linear. The more glucose in the blood, the more glucose goes into the kidney tubules. However, there's a point, see where this blue line is? Right about 180, and the units are in milligrams, 180, that we start to see a change. And if you take a look at this magenta line, this is reabsorption. Reabsorption is, although it's hard to see here, the reabsorption is occurring parallel to the filtration until we reach about 180, and then the reabsorption tends to level off, right? And this leveling off, of course, is due to the fact that we've basically saturated our carrier proteins, the SGLTs and the GLUTs, but primarily the SGLTs, so they can't keep reabsorbing anymore. And as the reabsorption occurs, we're starting to see the decrease in reabsorption as it starts to level off, whereas it was going up, it tends to level off. We're seeing a coordinated increase in excretion, right? As we're reabsorbing less, we're excreting more. So that's all, those all fit into play. And again, we'll have time to review this. Again, if you guys have any questions later, but that's kind of what's going on. Filtration, the rate of filtration of a certain substances stays the same, but what varies in this case is the reabsorption. The more a substance is, the higher the concentration, that reaches a point where it can't reabsorb anymore, and it starts to level off because it's saturating the carriers and it's tending to spill into the urine, thereby having what we call excretion. All right. All right, what about sodium and water balance? All right, so we've spoken about glucose, and we already know some aspects of sodium and water balance, especially, we spent a lot of time that first osmoregulatory lecture talking about movement of sodium into animals, movement of sodium out, water in, water out by a variety of different methods, right? Whether we're looking at the skin, whether we're looking at salt glands, right? Whether we're looking at gills, chloride cells, and of course the kidney in mammals. So how is sodium reabsorbed? And as I mentioned, as you saw in that table, most sodium is reabsorbed. Not all of it, whereas glucose is normally 100%, sodium is in the high 90s. So how is it reabsorbed? All right. Well, there's a couple different ways. I'm going to show you this first way here. Here is the kidney lumen, where the urine is, right? This is a kidney cell, and this is going to be ultimately where the blood is, right? Much like with glucose, there's two different transport mechanisms that have to be applied because that solute is going across two membranes. First, sodium, when it's entering into a kidney cell, it enters through, in this particular case, through facilitated diffusion down its electrochemical gradient. High concentration to low. This is going through ion channels. Once it's inside the cell, it's pumped out into the blood through primary active transport. And you remember that you've heard of the sodium-potassium ATPase. You've heard a lot about ATPases. This is an ATPase in which there's an exchange of sodium going, in this case, into the blood, and potassium going back into the cell. There's a lot of these different kind of opposing exchanges, much like when we're looking at the chloride shift, right, with chloride and bicarbonate. So facilitated diffusion coming in, even though it doesn't say it up there, it's facilitated diffusion through an ion channel coming in, into the cell, and then active transport going into the blood. Now that's one way. There is a second way, which if we go back to that original diagram we were looking at glucose transport, remember glucose is transported by this SGLT, sodium glucose link transporter. So sodium can also enter the cell by coupling with glucose. So sodium comes into the cell through sodium, so SGLT, and then it exits again through the sodium potassium ATPase. So this aspect is the same. Crossing the basolateral membrane is in the case. Same with both forms of sodium transport. It's at the apical membrane that it can either be SGLT or just facilitated diffusion of sodium. So because we know this, it's pretty fascinating, right? There's a lot of different tools that can be used in research as well as clinically to treat certain disorders. In some cases, whether we block the SGLT in diabetics or with sodium potassium ATPase in other cells, we can affect nerve function, muscle function. All right. So we've established the reabsorption of glucose and sodium. Now, of course, that's kind of normally how it occurs. Well, under normal circumstances, but there are certain factors that change. As we know, we spoke about homeostasis, where conditions change. We've learned a lot already about blood pressure, right? How we regulate blood pressure in terms of sympathetic, parasympathetic, remember regulating cardiac outputs, regulating peripheral resistance. One of the interesting things with the kidney is we know, as I mentioned before, the kidney is a very integral part on regulating cardiovascular function as well. And how does that work? Well, we've learned already how the kidney responds to high blood pressure. Remember we spoke about the myogenic response, or the constriction of the afferent arteriole, or also the tubuloglomerular feedback, where those macula densa cells sense high levels of sodium, respond by releasing adenosine, which also causes constriction of the afferent arterioles, reducing blood flow. Well, the kidney can also respond to low blood pressure. And the way it does that is through another group of cells called granular cells. And I'll show you a picture of those shortly. So these are distinct cells that are in the kidney. When the blood pressure drops, these granular cells produce an enzyme called renin. Renin is an enzyme that catalyzes the conversion of angiotensinogen into angiotensin 1. Angiotensin 1, once it gets into the blood, or once it's formed, it's immediately converted into angiotensin 2, which is the active, we'll call it a hormone. Now, it's converted into angiotensin 2 by an enzyme called ACE. Some of you guys that have worked in clinical settings or know of people who take blood pressure medication, you may have heard of ACE inhibitors. These are drugs that block the enzyme that makes angiotensin 2. Why do you want to block angiotensin 2? Because angiotensin 2 is going to increase blood pressure in a couple different ways. Number one, it can cause vasoconstriction of certain vessels, not the renal vessels, but some of the other ones. What does it do? It acts on the hypothalamus to increase the production of vasopressin. Vasopressin, we're going to talk about that shortly. This is actually a hormone that promotes the uptake of water to increase blood pressure. We also increase our thirst sensation because quite often if our blood pressure is low, it can be, that means our blood volume is low. So one way we can increase our blood volume is by drinking. So there are several different targets of angiotensin II. The blood vessels, the cardiovascular system. In this case, this would activate the sympathetic branch. It acts on the hypothalamus. And finally, which is what we're going to focus mostly on, it acts on the adrenal gland, the part of the adrenal gland known as the adrenal cortex. And we'll talk about this more in the endocrine system. But the adrenal cortex produces a hormone called aldosterone, which is a steroid, which promotes sodium uptake. We know from osmosis when sodium is moved in a particular direction, what follows? Water, right? So you bring up sodium, you bring up water, blood pressure is restored. So all of these are factors that are under the control of angiotensin, which initially is triggered by these granular cells in the kidney, which produce renin, which converts angiotensinogen into angiotensin 1. All right, so let's take a look at it. What are these granular cells? All right, so this is kind of a zoomed in picture. This is the Bowman's capsule. This is the glomerulus. It's kind of a large view, but you can see the podocytes. This is the afferent arteriole. This is the efferent arteriole. Notice the arrows, the way they're going. This is the distal tubule. This is the macula densa, which is not playing a role in this circumstance. What we're looking at are these purple cells. These are the granular cells. Some people call them juxtaglomerular cells. It's just a lot easier just calling them granular cells. These are the cells that make renin. All right. So when the blood pressure drops, activates these cells to make renin. What does renin do? As you know, renin enters into the blood, converts angiotensinogen, which by the way is produced by the liver, into angiotensin 1. Angiotensin 1 is converted by ACE into angiotensin 2. And then angiotensin 2 stimulates the release of aldosterone, which is a steroid hormone produced by the adrenal cortex. Now, granted, angiotensin 2 does those other things too, like I mentioned, right? So don't forget those. Oops, don't forget those. But right now we're talking about the effect on the adrenal cortex. All, of course, lead to an increase in blood pressure from that hypotensive condition. All right. So aldosterone, as I mentioned, increases sodium uptake. So how does it do it? There's always a mechanism, right? Physiology is all about mechanisms. So first of all, as I mentioned, it's a hormone produced from the adrenal cortex. It acts on the kidney tubules. How does it increase sodium uptake? It increases the number of sodium-potassium pumps on the basolateral membrane, increases the number of channels on the apical membrane. All right, so this is just writing, but if you see what it looks like here, here is the blood, right? Aldosterone released from the adrenal cortex enters, and aldosterone is a steroid. It's a lipophilic substance, so it crosses the membrane very easily, goes into the kidney cell, binds to a receptor, and what does it do? It activates two things. It increases the number of sodium and potassium channels, but sodium is the one we're focusing on right now, which allows sodium to move into the cell. And at the same time, it increases the activity of these sodium-potassium pumps, which once sodium is back in the cell, it moves sodium into the blood. So it's working kind of on both sides, right? Both sides of the membrane. Very effective way. And actually, there are certain medications for people who have high blood pressure that are used to block aldosterone. They call them aldosterone antagonists. So when you block aldosterone, what's going to happen? Sodium's going to go into the urine. Water is going to go into the urine because it flows that way, and thereby you're going to lower blood pressure. All right. So that's aldosterone. I want to talk about a couple other hormones that are very important in acting at the kidney to regulate blood volume. We know how aldosterone does it, right? Sodium uptake via channels and sodium-potassium pump. There's another hormone called ADH, and on the previous slide, it was referred to as vasopressin. You may have seen, remember this right here, vasopressin. Vasopressin is exactly the same thing as ADH. ADH stands for antidiuretic hormone. So let me just go for a second here. All right, so ADH equals antidiuretic hormone. Right. And with a name like that, many of you, just again from clinical experience or family or whatever, you know that diuretics, of course, their function is to make somebody pee more. Someone who has high blood pressure, right, makes them pee more. This is antidiuretic hormone. So antidiuretic hormone you would think would have the opposite effect. And in fact, that's exactly what it does. All right, so let me go back. Antidiuretic hormone acts on what are called water channels, called aquaporins. I like to think of it as like aquapores, like openings. Increases the activity of water channels, inserts them in the membrane. And of course, once water channels are inserted in the membrane, it allows more water to be reabsorbed by osmosis, essentially following sodium. And of course, sodium moved into the blood by way of aldosterone. All right, so let's take a look at a similar kind of diagram, except this is with ADH. So first of all, where does ADH come from? Again, this is something we'll talk more about in the endocrine system, but just to let you know now, ADH is produced in the hypothalamus. Comes down, also known as vasopressin, binds to receptors on the cell membrane of the kidney this time, and this activates a signaling mechanism, which I'm not going to have you worry about that, but that signaling mechanism increases the amount of what we call aquaporins, aquaporin 2, which is inserted into the membrane on the apical side. There are aquaporin 3s. Those are not regulated by ADH. They're always on the basolateral side. But of course, in order for water to move into the blood, you've got to have it coming in from the tubules as well. So ADH triggers the insertion of these water channels called aquaporins. Now water can enter into the cell and then back into the blood. The combined effect of uptake of sodium, uptake of water, increases blood volume, reestablishes blood pressure because we're responding to low blood pressure, low blood volume. All right. So what are some of the factors that affect vasopressin? Basically, all I really want you to know, there's a lot of stuff here. I want you to just focus on the fact that low blood pressure, low blood volume are the main factors that influence the production of vasopressin. Vasopressin isn't always produced. What activates it is decrease in blood volume, decrease in blood pressure, which activates the hypothalamus to produce it, which then allows for insertion of water pores. And of course, now we have increased water reabsorption, increased blood volume, increased blood pressure. So again, this is the whole homeostasis, right? We spoke about Homeostasis is when the response negates the stimulus. The stimulus is low blood pressure. The response is raising it. All right. I'm going to mention one other hormone that acts on the kidney. And in this case, we're looking at the opposite effect, opposite response. Remember, we just spoke about aldosterone, really angiotensin and aldosterone and ADH, all those A's, right? They respond when blood volume, blood pressure is low, right? What about, how does the kidney respond when blood volume is too high? I mean, we know, of course, how the autonomic nervous system works. We've already focused on that before, right? With sympathetic, parasympathetic. What about the kidney? Okay. There's a hormone called ANF. And I know at least one of you has worked in Dr. Glombotsky's lab, and for years he studied these peptides. ANF stands for atrial natriuretic factor, or atrial natriuretic peptide. Okay, and let me just spell that out for you. ANF equals atrial natriuretic. Factor, but I'm going to also put, it's more commonly known as atrial natriuretic peptide. Factor, it was called that because they didn't know what the molecule was. And there's other atrial factors as well. But their response is to respond to high blood pressure. And basically you can take those other two hormones, aldosterone, ADH, and just do the exact opposite, right? With ANF. What does ANF do? Basically, it inhibits vasopressin, ADH. It inhibits aldosterone. Ultimately, it decreases blood pressure and, of course, decreases renin, which is going to obviously lead to less aldosterone. So all these things, you can really think of it as they antagonize the response to angiotensin and aldosterone. And as a result, instead of promoting the uptake of sodium and water, we're excreting it. All right. So this is an old diagram, but sometimes old ones are good. I like this one because it really kind of summarizes those different hormones that we just spoke about and what are the conditions that trigger their effects. If we take a look, we have 4 conditions, right? We've got too much water, not enough water. Too much salt, not enough salt, all right? So let's take a look when we have a water deficit. So water deficit, again, we're looking at like low blood volume, right? In response to low blood volume, low water, what do? We produce vasopressin. And vasopressin promotes water uptake, right? When sodium is low, again, low blood pressure, low blood volume, we produce renin, which leads to this whole angiotensin pathway, which leads to aldosterone, which promotes sodium uptake. 2 different hormones there. What about when there's too much water and or too much salt? That's where ANF kicks in, right? And I should mention ANF is produced by the heart. I may have mentioned the previous diagram. So the heart is actually an endocrine organ in addition to its actions on pumping blood. Right? Very important endocrine organ. Excess water, excess salt leads to an increase in ANF, which is going to promote water uptake. Water excretion. Sorry about that. Water excretion and sodium excretion. So ANF promotes sodium and water excretion. So basically it's the same thing we saw in the previous diagrams, but just sort of showing it all in one picture. All right. Filtration, reabsorption, secretion. All right. I'm not going to mention much about secretion other than to say that this is where solutes that were not filtered across the filtration membrane at the glomerulus still end up in the kidney tubules by way of the peritubular capillaries. And so what enters into the kidney in this way? Well, different substances. This is typically where we see things like penicillin, I should say generally drug metabolites. These are some of the substances that are actively secreted as opposed to going through the normal filtration. Hydrogen ions, think of hydrogen ions, think of acidity, right? Choline. So these are substances that make their way into the kidney. I'm not going to go over the transport mechanisms of them, but just know that they enter into the kidney tubules, not from the glomerulus, but from the peritubular capillaries. All right. So one of the substances that can be actively secreted is hydrogen ions. So that brings us into discussing pH. And if we want to think of kind of at both sides of the coin, right? You can have a neutral pH or like normal physiological pH, or you can have the pH being too high or the pH being too low. So how does our body respond to that? Well, you probably have an idea that if our body is too acidic, we want to somehow get rid of that. If it's too basic, we want to get rid of that. So how do we do it? That's the question, right? Well, here we're looking at in the collecting duct. So this is where the collecting ducts play a very important role. There's two types of cells in the collecting ducts. There's what are called the type A intercalated cells and the type B intercalated cells. These are easy to remember, I think, in the sense that type A, think of A, these are cells that respond to if the blood is too acidic, think of A. Type B cells respond to conditions in which the blood is too basic. I'm not sure if that's how they came up with the letters, but it fits in pretty well, okay? All right, so very simply put, if our blood is too acidic, and of course we need a physiological pH because if we stray away from our normal pH, it affects enzyme functions, and it affects our general well-being. A lot of enzymes that normally break down substances or form substances, they're not going to work properly. Digestion's not going to work. A lot of different things, right? Muscle function. So how do we respond to acidotic condition? Basically what happens, what we do when the conditions are acidotic, and I'm just going to really cut to the chase here, is hydrogen is excreted via active transport. Called it like basically it's like a proton pump. Hydrogen is excreted out through, and where is excreted? It's excreted into the kidney tubule. Right now we're getting rid of hydrogen ions, so that hopefully will reestablish the pH. What about the other conditions, right? There's other stuff going on. I just want you to focus on the hydrogen ions. If the conditions are too basic, right? Bicarbonate, sodium bicarbonate has an alkaline activity. It helps to buffer things, right? Help bring the pH back to more of a physiological level. But if we have too much of it, we need to get rid of it. So with these type B cells, they primarily excrete bicarbonate. Okay, so let's focus on this for a second. The function of type A cells is to respond to acidic conditions by pumping out hydrogen through active transport in exchange for potassium. It's what we call a proton pump. Type B cells excrete bicarbonate in exchange for chloride. And this might look familiar to you. This is another chloride shift. Remember we saw the chloride shift with red blood cells, with getting rid of bicarbonate in exchange for chloride. We see it here. We'll also see it in the digestive system. All right. Okay, so we're kind of on the final frontier here. Let's see how we're doing. We are plugging on. Okay. All right. So to kind of bring things full circle, right, we started off with blood entering into the glomerulus, being filtered, right? About 99% of what's filtered, for the most, except for urea, but most of it is reabsorbed. What is not filtered to begin with, things like potassium, choline, creatine, and other drug metabolites are secreted. And the volume of urine that's excreted is basically dependent upon the amount that's filtered, subtracting what's reabsorbed, adding what's secreted, and that ends up being our volume of excretion. All right. We've already seen this diagram, right? This is basically showing filtration, secretion, reabsorption, and you can see right here if a substance is filtered, if 12 millimole, if 3 millimoles are secreted into it, and 9 millimoles, I'm sorry, if 6 millimoles are reabsorbed, that's 12 plus 3 minus 6, we end up with 9. That's going to be excreted. All right, so I'm going to go through just maybe one or two more little equations. I'm not going to have you calculating anything. I just want you to know just kind of some of the ways of measuring urine function. One is called clearance, renal clearance. It's the volume of plasma from which a substance has been removed by kidneys per unit time. So the volume of the plasma that contains the amount of a substance that's been excreted. This is calculated by the excretion rate over the plasma concentration. Now, we haven't gone over excretion rate, but another way to calculate it is basically taking the GFR, multiplying it by the plasma concentration of a substance. And you might think this is a typo. It looks like insulin. Inulin is a substance that is freely filtered, does not get reabsorbed or secreted, and it's a perfect kind of a substance that's used to understand what GFR is. So in other words, glucose is a good substance to study absorption because it's 100% reabsorbed, right? Inulin is a good substance to study basically filtration and clearance because it's filtered, no more is added, no more it's reabsorbed. So basically what's filtered is equal to what is cleared. All right. So if we take a look here, we start off with 0.5 millimole of inulin. That's the filtration rate, the GFR. And we end up excreting 0.5 millimole of inulin. So that's basically what we're talking about. So the clearance of inulin is exactly the same as the GFR. That's one way to look at it. So inulin clearance equals GFR. What goes in is exactly what is cleared, what goes out. Now, what about, and I mentioned the value of that in this case, and so the clearance of inulin is exactly the same as GFR. What about the clearance of glucose? Well, remember, glucose is a substance that's filtered, but it's completely reabsorbed, right? It's completely reabsorbed. Whereas, remember, inulin, it's filtered, nothing is reabsorbed, nothing is secreted. So what goes in is exactly what comes out. With glucose, what goes in is whatever the concentration is, what goes out is 0. So the clearance rate for glucose is 0 because no glucose is excreted in the urine. All right, and you can see that right here. Say, for example, the clearance of glucose, 125 milligram. And again, these are just random numbers. 125 milligrams of glucose are filtered, right? It's all reabsorbed. So we have 0 left. So just to kind of do a comparison, the clearance of glucose is 0 because it's all reabsorbed. None of it's cleared through the urine. The clearance of inulin is exactly the same as GFR because what goes in is exactly the same as what goes out, and there's no secretion and reabsorption. All right. So determining the fate of solutes and renal tubules. If the clearance of a substance, and that's what CX is, if the clearance is greater, well, let me look at the second one first. If the clearance rate is less than the GFR, then the substance was reabsorbed, right? So we know with glucose, the clearance is 0. That means, and started off with GFR, so it's completely reabsorbed. If the clearance is greater than the GFR, that means you've had some stuff added to it, so that means there is some secretion. So in other words, just to go back here for a second, let's see. Let's go back here. So saying if glucose, we filtered it, instead of it all being reabsorbed, say if none of it was reabsorbed, but instead we had more added to it, that would mean that our excretion rate, maybe say if we added 2 milligrams, our excretion or clearance would have been 127. That's greater than 125, so that would mean that there was secretion. All right. And just to show you some of these values, again, I'm not going to have you do any calculations, but taking a look at some of them we looked at, glucose, 0 clearance rate, it's all reabsorbed. Inulin clearance rate is the same as the GFR because there's no reabsorption or secretion. And we didn't look at these substances, but here's a good example of creatine and PAH, 2 other substances that are used to study. Notice their clearance rate is greater than the GFR, which is normally 125. So that means there's active secretion of these substances. All right. So we're on the final frontier, guys, finally, right? We've made it out of the kidney. We formed the urine, right? We're excreting. And of course, what happens? The urine travels, once it leaves the collecting duct, it leaves what's called the renal pelvis, travels into the ureter, and leads to the bladder. And the bladder stores the urine until it's excreted. The excretion or urination is also known as micturition. All right, so this is a nice kind of a frontal view of the structures that play a role. Here we've got the 2 ureters. We've got the bladder. Here we've got part of this is the urethra, which is going to lead out to the outside world. But I wanted to point out a couple key structures. Number 1 is this muscle in the bladder that's known as the detrusor muscle. Very thick, smooth muscle. I also want to point out a couple different circular muscles called sphincters. We have what's known as the internal urethral sphincter and the external urethral sphincter. And whether these sphincters are open or closed determines our ability to urinate. So we'll take a look at how this is controlled. All right. So say our bladder is full, right? Well, when our bladder is full, what happens is this causes the wall of the bladder, those detrusor muscles, to stretch. When the detrusor muscle stretches, there's specialized nerve endings called stretch receptors that are activated, much like you've heard of chemoreceptors, right? Baroreceptors for pressure. These are receptors or specialized nerve endings that respond to stretching. When they're stretched, a couple things happen. Number 1, we see an increase in the activity of the parasympathetic system and a decrease in sympathetic, right? So the process of urination, if we talk about the autonomic nervous system, we see an increase in parasympathetic, decrease in sympathetic. All right, so what does this mean? This triggers contraction of the detrusor muscle. Contraction of the detrusor muscle, which causes that bladder now to contract, right? It's like we want to squeeze out the urine. But the urine's not going to go anywhere because the sphincters are closed. That the same activation of the parasympathetic system also triggers an opening of the sphincter, internal sphincters. Let me go back for a second. So this is normally closed. When the parasympathetic comes down, it causes detrusor to contract. It causes this sphincter to basically open up or relax. And that's fine, but it's not the end of the story, because remember, we have two sphincters. At the same time this is going on, the somatic branch of the peripheral nervous system, remember this is the voluntary. It's like when we talk about acetylcholine, right? Neuromuscular junction. This triggers relaxation of the external urethral sphincter. Here's internal, here's external. When that sphincter relaxes, that allows it to open. So think of relaxing of the sphincters allows them to open, since they're normally closed. The contraction of the detrusor, the opening of the internal, followed by the voluntary opening up of the external, allows us to urinate. And of course, this right here is a learned response, right? This is something, if you talk about kids potty training, right? And once they have to go to the bathroom, there's no like waiting, right? When they go, they go. But it's with a learned response that we're normally able to keep that sphincter closed until we're near a rest stop, right? Or near a bathroom. I probably shouldn't have put in this in there, but just to kind of, there's a lot of extra stuff you don't need, but just to kind of illustrate, this is where the stretch receptors are. It sends a signal up to the brain, up the spinal cord to the brain, which then activates the parasympathetic, which causes contraction of the detrusor, relaxation of the internal sphincter, and at the same time, the somatic branch opens up the external. Not the best diagram, but hopefully it helps. All right, so you can say that micturition is under dual neural control. The parasympathetic controls the internal urethral sphincter, that's IUS, as well as the detrusor. I should have put that in there. And the somatic branch of the peripheral nervous system, this controls the external urethral sphincter. So it's kind of a cooperative response by both the autonomic and the somatic. All right. Last thing I wanted to mention is, so we talk about how medicines have been developed once we know the receptor, once we know a channel. I mentioned about diabetes with SGLT. Well, there's a condition called incontinence. Some of you might be familiar with it, just family or clinical setting. This is where people basically cannot control the ability, the need to urinate. It happens a lot to elderly people. Not every elderly person, but a lot of them. If someone has like neurological injury, quite often if they have the sensation to urinate, they have to be near a bathroom right then and there, as opposed to when you're healthy, typically you can wait until you can go. So there's a drug called tolterodine that, what does it do? This basically blocks the ability of the autonomic neurons from stimulating contraction of the detrusor muscle. So remember, the detrusor muscle contracts, right? And that allows us to urinate. Well, the problem with people that are incontinent is that it's kind of out of control. There's spasms and they have difficulty controlling their urination. This actually slows down the contractions Doesn't completely eliminate them because you don't want it to be eliminated because you want them to have the sensation that they have to go to the bathroom, but at least reduces the urgency. So, all right. That should be the end of that. So, all right. So that takes us to the end of the renal system. Any questions? I know everyone's usually pretty quiet and hiding. Oh, I should look if there's anything on the chat board. Nothing. Okay. All right. So if there's no other questions, tomorrow we'll start the endocrine system, and I will be posting something later today. So in the meantime, everybody have a great day, and I'll see you tomorrow.